All right, welcome to the Explainer. Today, we're going to crack open a scientific mystery that's hidden deep inside the brain. It's basically a detective story, but at the scale of a single neuron, where one tiny electrical clue reveals the secret behind our crazy fast hearing.
Okay, so think about this for a second. When you hear a sound, you pretty much instantly know where it came from, right? Well, that ability hinges on your brain detecting these tiny sub-millisecond differences in when the sound hits each of your ears. The neurons that do this have to fire with just unbelievable speed and reliability. So, how in the world do they pull that off?
Well, to answer that, we've got to zoom in on a very special connection in the auditory brainstem. That's the part of your brain that's first in line to process sound. And it's right here that scientists found this really strange electrical signature, a mystery that just had to be solved. So, meet our crime scene, the calyx of held synapse. I mean, this thing is one of the largest and fastest connections in the entire nervous system. It's a true high-performance engine for hearing. It's basically built for speed. But yeah, it has a secret. When researchers recorded the electrical activity from the synapse, they saw what you'd expect, right? This massive spike, the action potential. But then, right after it, there was something else. A weird, secondary bump. You can actually see it right there on the trace. A little electrical echo that scientists have a name for, a depolarizing after potential, or DAP for short.
And that little bump? Well, that's our whole mystery. Was it just a random glitch in the system? Some insignificant electrical quirk? Or was it actually there for a reason, playing a crucial role in this whole high-speed circuit? That's what we're going to find out.
Okay, so let's start the investigation. Let's follow the clues. First things first, the researchers had to figure out what was even causing this echo. Like which specific little molecular gates, the ion channels, were responsible for it. So their first tool of choice was this poison called tetrodotoxin. You might have heard of it as TTX. It's the stuff that's famously found in pufferfish, and its superpower, if you can call it that, is that it specifically blocks voltage-gated sodium channels, the exact channels that make the main action potential spike happen. And boom. This gave them their first major clue. I mean, just look at the slide here. When they put just a tiny bit of TTX on there, the main spike was barely touched. But that mysterious echo? It just vanished. This was a huge lead, pointing straight at a sodium current being the cause. Okay, so to be 100% sure, they used a different sodium channel blocker. This one's called QX314. But this time, they put it inside the neuron itself, and guess what? The result was exactly the same. The echo disappeared. So that sealed it. The culprit was definitely a local sodium channel, right there at the synapse. All right, so big breakthrough for the investigation. This echo isn't just some passive rebound. This thing is an active signal driven by sodium flowing into the neuron after the main spike. But that just led to the next big question. Which specific type of sodium current was doing it? All right, let's get into the next phase and meet our prime suspects. Because here's the thing. Not all sodium currents are the same, and the researchers had managed to narrow it down to two main possibilities. So, on one hand, you've got the persistent current, or INAP. Just think of it like a slow, leaky faucet of sodium. The other one, though, the resurgent current, INR, it's way weirder. It actually activates after the main spike is over, briefly reopening to let more sodium in before it finally shuts down. I mean, that is just a really, really strange way to behave. So, here's the real problem for our detectives. There are no drugs that can block one of these suspects without also blocking the other. It's a classic whodunit problem, right? With no way to pharmacologically isolate the culprit, how do you finger the right one? Well, they did something brilliant. They turned to a super powerful tool, a digital twin. They built this incredibly detailed computer model of the calyx of Held, programming in every little electrical property they knew about. Once the model could perfectly replicate what the real neuron was doing, including that mysterious echo, they could do their investigation virtually, just removing one suspect at a time to see what happened. And here we go. This is the smoking gun. In their digital model, when they took out that leaky persistent current, the echo was still there. But, get this, when they remove the resurgent current, INAR, the echo is just gone, completely gone. That was it. That was the definitive proof they needed. And just like that, case closed. The computer model proved it, really, beyond a reasonable doubt, that the resurgent sodium current was the one and only source of that mysterious electrical echo. But, finding the culprit is only half the story. The real question now was, why? What was its motive? Because in this story, our culprit, it isn't a villain at all. Turns out it's a hero with a really important job. So researchers started looking at how this echo changes as a baby rat gets older. And what they found was, wow, it was striking. In the really young animals, before their hearing is fully up and running, the echo is pretty small and slow. But as the animal gets older and hearing comes online, that echo gets bigger, faster, and way more obvious. And this lined up perfectly with the neuron's new ability to fire at these insane speeds. So naturally, this led them to a really powerful idea. This resurgent current and its little echo, it's not a bug, it's a feature. It's basically a developmental upgrade that lets these neurons fire way faster and more reliably, which is exactly what you need to pinpoint where a sound is coming from.
But hey, a good detective has to prove their theory, right? So they designed this one last absolutely brilliant experiment. They took an immature neuron, one that normally struggles to keep up with high-frequency signals, and they used a special peptide to artificially give it the strong resurgent current that only the mature neurons usually have.
And the result? You can see it right here on this graph. It was just stunning. Before they added the peptide, the immature neuron had about a 92% success rate firing at high speed, which is pretty good, but it's not perfect. But after giving it that artificial boost of resurgent current, well, its success rate shot up to a perfect, flawless 100%. I mean, come on, case closed. For real this time. So, what's the big takeaway here? That weird little echo, it's like a biological turbocharger for the neuron. It gives the neuron this extra electrical kick after it fires, helping it reset and get ready to go again way faster. It's the clever little trick that lets this synapse keep up with the insane demands of processing sound. This whole detective story, from a tiny electrical bump all the way to a fundamental mechanism for hearing, it just shows you how elegantly our brains are put together. And it really leaves you with one final thought, doesn't it? What other incredible hidden mechanisms, what other elegant solutions for high-speed computing are still just waiting there to be discovered right inside our own heads?